Good morning. I am Fabiana Carletto Korber. I am part of a research team made up of professionals from different disciplines from the National University of Córdoba-Argentina, being the main theme of SciTech Central Dentistry 2021, exploring advances in the field of dentistry. I am going to tell you about genetic variability of Streptococcus mutans and its association with caries. In this slide, we can observe where Cordoba city is located in Argentina. It also shows the photo of the building called Pediatric Argentina at the university campus where the Faculty of Dentistry is situated. In another picture, you can also see the building of the Faculty of Exact, Physical and Natural Sciences, where this study was carried out. Before I start the presentation, I want to say that none of the authors present conflicts of interest. This study was approved by the Institutional Ethics Committee of the Dentistry Department. It was supported by a grant of the Secretaría de Ciencia y Técnica of the National University of Córdoba. Genetic structure of pathogenic organisms. The appearance of new diseases, or the reemergence of others, reveal changes in the epidemiology of pathogenic organisms. Despite numerous technological advances, in many cases, it has not been possible to explain the mechanisms how microorganisms cause diseases. The factors that determine the emergence of pathogenic bacteria, why certain bacterial clones are pathogenic and others are not, or why non-pathogenic strains possess genes associated with virulence. Although causes are multifactorial, population genetic studies can contribute to the understanding of the mechanisms involved, providing information about the nature of the existing genetic variation of natural populations. Bacteria exhibit rapid evolutionary changes due to their short generation times and their enormous population sizes. Variations in the population genetic composition depend on processes such as mutation, recombination, migration, genetic drift, and natural selection. For this reason, studies on genetic structure and population dynamics try to explain the mechanisms by which a species evolves, and also answer specific questions about the history of their populations. Globally, this kind of studies in bacteria have detected three types of structures: clonal, recombinant, and epidemic. Dental caries is one of the most widespread diseases in the world, and is frequently diagnosed in children. It is associated with different bacteria that should be studied with a population genetic approach, being Streptococcus mutans the one commonly associated with dental caries. In the following slides, I will characterize the Streptococcus mutans bacteria and how the biofilm is developed, which is bound naturally on teeth and is also implicated in dental caries. This disease is associated with shifts in the microbial balance of the biofilm, resulting in increased proportions of acid-producing and acid-tolerating bacteria, especially but not exclusively mutant streptococci and lactobacilli. The broad composition of the dental biofilm has been explored. Glucan comprises 10 to 20% dry weight of dental plaque and fructan approximately 1 to 2%. Clearly, these proportions will vary depending in part on the duration since the last intake of food. Dental biofilm also contains approximately 40% of dry weight proteins, mostly derived from bacteria and saliva, plus variable amounts of lipids, calcium, phosphorus, magnesium, and fluorine compared with surrounding saliva, being also approximately 80% water. The ecological biofilm hypothesis proposes that dental caries are the consequence of changes in the natural balance of the oral microbiota that occurred as a reaction to an alteration in the conditions of the oral environment, leading to an increase in the proportion of acidogenic and aciduric bacteria. The cariogenic potential of Streptococcus mutans is attributed to virulence factors, such as the ability to metabolize carbohydrates as they produce acid like lactate acidogenesase, which is their main source of energy in anaerobiosis. This figure shows the revised model of glucosyltransferase glucan-mediated bacterial adherence and cariogenic biofilm development, originally proposed by Rotlin and collaborators in 1983. The glucosyltransferases secreted by Streptococcus mutants are incorporated into the pellicle, particularly the one known as GTFC, and adsorbed on bacterial surfaces, mainly GTFC, including microorganisms that do not produce glucosyltransferases, for example, actinomyces. Furthermore, salivary alpha amylase is also included into the pellicle, which can also bind to glucosyltransferases. Surface-adsorbed GTFC and GTFC rapidly utilize dietary sucrose to synthesize insoluble and soluble glucans in situ. The soluble glucans formed by GTFB could serve as primers for GTFB, enhancing the overall synthesis of exopolysaccharide. Concomitantly, starch is digested by amylase, releasing maltose and a myriad of oligosaccharide, which can be incorporated into the polymer molecule through acceptor reactions, particularly by surface-adsorbed glucosyltransferase B. The glucosyltransferases adsorbed onto enamel and microbial surfaces provide in situ an insoluble matrix for dental biofilm. The glucan molecules provide avid binding sites on surfaces for Streptococcus mutans and other microorganisms, mediating tight bacterial clustering and adherence to the tooth enamel. In addition, glucosyltransferases adsorbed bacteria become the glucan producers, binding to tooth and microbial surfaces by the same mechanisms. This model could explain the rapid formation and accumulation of highly cohesive adherent biofilm in the presence of sucrose and possibly starch, even if the number of Streptococcus mutants is relatively lower. After the establishment of a glucan-rich biofilm matrix, ecological pressure, for example pH, will determine which bacteria may survive and dominate within biofilm under frequent sucrose or other fermentable carbohydrate exposure. Other characteristics are that in the cell wall there are proteins that play an important role in adhesion, aggregation and coaggregation phenomena, as well as polysaccharides such as granulose glucose polysaccharide that display specific antigens. Various polysaccharides and proteins have been identified to characterize different bacterial serotypes. Therefore, serotype C, E, F, and K have been associated with Streptococcus mutants, whereas serotypes D and G have been identified in Streptococcus subrenus. Among the characteristics of Streptococcus mutants genotypes, the following stand out: high diversity number of genotypes in a certain population, homogeneity number of shared genotypes in a certain population, and stability persistence of genotypes throughout time in a certain population. Children tend to have fewer genotypes, less diversity compared to adults, suggesting that new strains of this bacterial species are acquired over the years. In the last decades, the increase in population genetic studies has contributed to elucidating important questions about the evolution of the pathogenesis of bacteria with clinical interest. Thus, the objective of this study is to revise and update concepts of the last 15 years related to the geographical distribution of the different genetic variants of Streptococcus mutants and their association with dental caries. To address this issue, we had to summarize the knowledge about the geographical distribution of the different genetic variants of Streptococcus mutants using biochemical methods. Approximately 66 to 80% of the strains present in the oral cavity worldwide were classified as serotype C, followed by E and F. Knowing the relative proportion of each serotype is important, because serotype C is detected more frequently in healthy patients, while non-C serotypes, or A mixture of multiple serotypes, are more frequent in subjects affected by heart disease or those who underwent surgery. Different molecular studies show that the most frequently observed is serotype variant C. For example, the study of Shibata and co-authors included preschool children from Fukuoka, Japan. The study detected serotype C in 84.8% of the strains. In other study in Japan, but in the city of Osaka, which included 69 healthy children, serotype C was obtained in 75% of the strains. In Bangkok, Thailand, in a simple group comprised of children and adults, serotype C showed the frequency of 70%. Besides, in Finland, serotype C was detected in a frequency of 41.6%, but the study included a very low sample size. And finally, in Argentina, in our study, which was carried out on healthy children attending 4 different schools in the Córdoba province, Argentina, we detected serotype C in 53.2% of the strains. Regarding our studies, Figure 1 shows the serotypes that were identified on healthy children attending 4 different schools in Córdoba province, Argentina. This study was the first analysis of the circulating serotypes in Córdoba, one of the most populated provinces in Argentina. In addition, we analyzed which serotypes were associated with greater caries prevalence in the population. Non-significant differences were found when considering the mean number of caries for the different serotypes. As a result, the idea that non-C serotypes were associated with a greater health risk could not be completely confirmed. However, Figure 2 shows that the sick population, thus, the third of the population with the highest caries scores, exhibited differences in the serotype composition compared with the non-sick population. In the sick population, serotype K was absent, and the presence of serotype C was significantly lower. Furthermore, serotype K was significantly associated with the non-sick population, the population with healthier oral cavity conditions. In another study performed by our research group, we considered that one of the interesting questions to clarify is the type of genetic structure of the populations of Streptococcus mutans. Is it recombinant or clonal? We know that most living organisms have the ability to reproduce asexually and sexually, although bacteria can also acquire genes or genetic variation through lateral gene transfer. The latter two mechanisms are sources of recombination that can create mosaic genomes, which may violate the assumptions of tree-like evolution. Previous studies on Streptococcus mutants using multi-locus sequence typing schemes have drawn different conclusions about the population genetic structure of this bacterium. Nakano and co-authors in 2007, using strains from Japan and Finland, and Fabiana and collaborators in 2011, using strains from Thailand, detected a clonal structure, whereas Du and co-authors in 2010, using strains with a worldwide distribution, identified recombination as the main force shaping the genetic structure. In the study of Gonzalez et al. In 2017, we used several statistical approaches to reveal the population genetic recombinant structure, using sequences generated by us from Argentina combined with those of Nakano and Fabiana. In our study in 2017, we found a high number of alternative connections among the 137 sequence types in the NeighborNet graph computed with split trees. If you look at the colors, there are no haplotype groups that correspond to any of the countries considered. In general, the sequence types are all intermingled. Few strains are shared between countries. According to the multi-locus sequence typing scheme and LSD used in this study, most of our statistical supported the existence of recombination at the intergene level. Besides, we detected that the genetic structure may be different in each country. We also identified low levels of recombination in strains of Streptococcus mutants from Japan, Thailand, and Finland, similar to that detected by Nakano and collaborators in 2007 and Fabiana and co-authors in 2011. In contrast, there were very high levels of recombination in strains from Argentina. Therefore, it is not surprising that, in the Gonzalez et al. Study in 2017, as well as in that of Du and collaborators in 2010, the different trees obtained with Everest and STAR2 showed different sequence type clusters, all with very low statistical support. The incongruence length difference test revealed homogeneity conflicts between genes, which may have been produced by recombination. In the same way, the Bayesian analysis performed in the present study using structure version 2.3.4 detected that 45 strains could have admixed ancestry as the product of recombination among strains with high membership in clusters 1, 2, and 3. Considering the model proposed by Maynard Smith and collaborators in 1993, our global results support the population structure of Streptococcus mutants. in which recombination or genetic shuffling might be the most important evolutionary process occurring in this bacteria. Nevertheless, it must be considered that recombination levels can vary between countries, as observed in our study, and this could explain the differences observed by the different authors, as was already explained. In summary, the results of our study are mostly consistent with the idea that recombination is an important force leading the evolutionary history of Streptococcus mutans, thus supporting the results of Waterhouse and Russell in 2006, Magrueva and collaborators in 2009, Du and co-authors in 2010, and Cornetto and collaborators in 2013. In our study, we also detected that many of the 137 sequence types from 193 strains of the 4 countries analyzed showed very low nucleotide differences among Streptococcus mutans. Many sequence types were exclusive to each country, and the festive values, which measured both differences in the nucleotide composition among sequence types and their frequencies between pairs of populations, showed significant differences between Argentina versus Japan and Argentina versus Thailand, 0.022 and 0.035 respectively. Of these 137 sequence types, only 12 were shared between 2 or more countries. These results would indicate that there are low levels of current levels of gene flow among countries. This result is compatible with a scenario of population homogenizing due to a recent range expansion of Streptococcus mutans, and a subsequent differentiation with moderate to low levels of current gene flow. Related to this, another question is to understand how the expansion of Streptococcus mutans would have occurred. This screen shows the dietary changes in human history. The importance of commensal microbes for human health is increasingly recognized, yet the impacts of evolutionary changes in human diet and culture non-commensal microbiota remain almost unknown. 2 of the greatest dietary shifts in human evolution involved the adoption of carbohydrate-rich Neolithic diets. One includes farming, beginning near 10,000 years before Preventive, and the other, the more recent advent of industrially processed flour and sugar, approximately in 1850. Adler and collaborators in 2013 showed that dental calculus is ubiquitous in both present-day and ancient human populations, and microscopic analysis has shown that it accurately preserves bacterial morphology over millennia. Calculus represents one of the few sources of preserved human and hominid microbiota, and genetic analysis has the potential to create a powerful new record of past dietary impacts, health changes, and oral pathogen genomic evolution deep into the past. The temporal transect of ancient dental calculus samples provides the first views about the timing and nature of changes in human oral bacterial composition and diversity over the last 7,500 years. The composition of oral microbiota underwent a distinct shift with the introduction of farming in the early Neolithic, with the earlier hunter-gatherer groups displaying fewer caries and periodontal disease-associated taxa. This is consistent with skeletal evidence showing marked increases in periodontal disease following the transition to an agricultural diet, suggesting a major impact on the human oral ecosystem around this time. This is thought to be caused by increased amounts of soft carbohydrate foods compared with hunter-gatherer diets. After the transition to agriculture in the early Neolithic, there was a notable consistency in the composition of bacteria through the medieval period, about 400 years BC. In parallel with the broad similarity of food processing technologies during these times. In contrast, today's oral environment is much less biodiverse and dominated by potentially cariogenic bacteria. According to Adler, the demographic expansion of Streptococcus mutans may have occurred due to 2 important dietary changes. The first involved a carbohydrate-rich diet associated with agriculture, while the second one was associated with the recent development of industrially processed flour and sugar. Caries and periodontal disease are both polymicrobial, flab-mediated infections, thought to result from perturbation of a healthy, ecologically balanced oral biofilm that can occur from dietary changes, such as the increased consumption of fermentable carbohydrates. Caries has become a major endemic disease affecting 60 to 90% of school-aged children in industrialized countries, while periodontal disease occurs in 5 to 20% of the adult worldwide population. Notably, oral bacteria are also associated with many systemic diseases, including arthritis, cardiovascular disease, and diabetes, in addition to diseases of the oral cavity. On the other hand, both Carletto and co-authors, and Gonzalez and collaborators, recognize a single stage of Streptococcus mutans demographic expansion related to the establishment and worldwide development of agriculture. This screen shows the results of the extended Bayesian skyline plot analysis. The y-axis represents the effective population size and the x-axis represents time. The black lines show the median values according to this. The expansion of Streptococcus mutans began about 5,000 years ago, from a small number of ancestral strains. This time estimation is within the 95% confidence interval of Carletto and co-authors that estimated the expansion in 10,000 years ago. The population genetic structure of Streptococcus mutans in Argentina is highly recombinant, which reflects the largest waves of human immigration that occurred in the 19th and 20th centuries. On the other hand, demographic analysis suggests that these bacteria experienced a population expansion that coincided with the beginning of agricultural development 10 to 5,000 years ago. This situation requires that several challenges be addressed to ensure accurate and quality diagnostics. In this sense, it is important to instruct and educate future health professionals about these types of techniques, indicating their advantages and limitations. The idea is to promote the development of interdisciplinary teams that, in the future, could design, implement, standardize, control, and interpret these diagnostic tools. Before ending this conference, I would like to present you to the people who have worked in this line of research since the 2000s. I would also like to thank them for their collaboration in the design of this presentation.